In today's lecture, we'll be talking about the metabolism of chylomicrons, also referred to as the exogenous lipoprotein pathway. Chylomicrons ensure that fats from our diet are absorbed and transported to the various cells where they are needed. The liver excretes bile through the bile duct into the small intestine, and this bile contains several components or several constituents, which includes cholesterol, conjugated bilirubin, phospholipids, bile salts, and then several other components. So these are basically some of the most important components that are contained in the bile or present in the bile that are excreted by the liver through the bile duct into the small intestine. When we ingest diet rich in fats, such as cholesterol and triglycerides, these fats get into the small intestine. On getting into the small intestine, these fats, specifically triglycerides, is acted upon by bile salts. So what bile salt does is that it acts as a detergent by hydrolyzing the triglycerides and converting into fat droplets. Fat droplets. After the action of bile salts on the triglycerides and converting into fat droplets, then the pancreas also secretes an enzyme called pancreatic lipase. Pancreatic lipase. So, what this enzyme does is, after the action of bile salts on triglycerides, in which it converts triglycerides to fat droplets, the pancreatic lipase now have or gain access to the bonds connecting the fats together and break those bonds. When these bonds are broken, it results in the formation of free fatty acids and monoglycerides. So the action of pancreatic lipase on the fat droplets will convert these fat droplets of triglycerides into free fatty acids and monoglycerides. When that occurs, now basically this occurs in the intestinal lumen. After free fatty acids and monoglycerides are obtained, these molecules, free fatty acids and monoglycerides, as well as the cholesterol obtained from the diet, are then absorbed by the intestinal mucosal cells, also referred to as enterocytes. Now, in the enterocyte, there is rough endoplasmic reticulum and there is smooth endoplasmic reticulum. Now, in the small endoplasmic reticulum, there is a synthesis of a type of protein called apolipoprotein B48, also referred to as ApoB48. Also, in the smooth endoplasmic reticulum, there is a process of esterification. So, these molecules that are absorbed, free fatty acids, monoglycerides, and cholesterol, will be esterified, and that will result in the formation of triglycerides and cholesterol esters. That is to say, triglycerides will be reformed in the smooth endoplasmic reticulum, and then cholesterol will be esterified to form cholesterol esters. So TG represents triglycerides, as is written here. TG represents triglyceride and CE represents cholesterol esters. So in the smooth endoplasmic reticulum, triglycerides and cholesterol esters will be formed. Now, when this is formed, there is a protein called microsomal triglyceride transfer protein. So, still in the enterocyte, there is a protein called microsomal triglyceride transfer protein. What this protein does is it ensures that This ApoB48 is bound to the triglyceride and the cholesterol esters together with phospholipids. Remember, I stated that in the bile, phospholipids are also present. And so these phospholipids will also form as part of the chylomicrons. So this protein ensures that triglycerides, cholesterol esters, phospholipids, the free cholesterol, and this ApoB48 are bound together. And that will result in the formation of this spherical structure where triglyceride and cholesterol will be in the core of the molecule. And then the outer shell of this structure will contain phospholipids and free cholesterol together with this ApoB48. Now, this is the chylomicron particle. The function of ApoB48 is that it ensures the structural integrity of the chylomicron. When this is formed, this chylomicron particle directly enters into the lymphatics. Normally, nutrients absorbed from the small intestine goes into the portal circulation, where they are first carried to the liver. But in this case, this chylomicron particle does not go into the portal circulation. It goes directly into the lymphatics, into the intestinal lymphatics. And then through the thoracic duct, it goes into the systemic circulation. In the systemic circulation, this chylomicron particle is not mature. So at that point, it is called the nascent chylomicron. But then, this chylomicron, this nascent chylomicron, associates with high-density lipoprotein. And high-density lipoprotein contains two apoproteins, apoprotein E or ApoE and ApoC2. So what the HDL particle does is, by association with nascent chylomicron, it transfers these apoproteins to the nascent chylomicron. You can see there is a transfer of this ApoE to this nascent chylomicron and transfer of ApoC2 to this nascent chylomicron. And when these two apoproteins have been transferred to the nascent chylomicron, it will result in the formation of the mature chylomicron. So these mature chylomicrons now contains the ApoE, ApoC2, and ApoB48. This mature chylomicron is carried from the blood to various peripheral tissues such as the adipose tissue, the skeletal muscle, and the cardiac muscle. And then when we ingest diet rich in fat, insulin is usually produced from the beta cells of the pancreas. Now, what this insulin does is it stimulates an upregulation of an enzyme called lipoprotein lipase in these tissues, in the adipose tissue, in the skeletal muscle, and the cardiac muscle. And these lipoprotein lipase are released from the cells into the capillary, okay, into the capillary of these tissues. Where the lipoprotein lipase is located is at the wall of the capillaries. So at the wall of the adipose, capillaries of the adipose tissue, skeletal muscle, and cardiac muscle, that is where lipoprotein lipase is located. And then this mature chylomicron will have to pass through the capillary. Now, in trying to pass, once it gets into the capillaries, as a result of the presence of ApoC2, this lipoprotein lipase will be activated. So ApoC2 activates lipoprotein lipase or stimulates the activation of lipoprotein lipase. And once lipoprotein lipase is activated, lipoprotein lipase will hydrolyze triglycerides. So lipoprotein lipase will break down triglycerides to form free fatty acids and glycerol. If this hydrolysis occurs in the capillary wall, in the capillary of the adipose tissue, the adipose tissue will absorb the free fatty acids that are produced and store it. But then if it occurs in the skeletal or the cardiac muscle, these free fatty acids will be used to produce energy or to generate energy. And that is because these free fatty acids will enter the beta oxidation pathway. So both in the skeletal muscle and cardiac muscle. These free fatty acids that are produced will undergo or pass through the beta oxidation pathway to produce acetyl-CoA. Now, this acetyl-CoA will now enter the Krebs cycle, also called the tricarboxylic acid cycle. Now, when acetyl-CoA enters the Krebs cycle, it will now produce what? ATP, which is used to generate energy. So, the skeletal muscle and the cardiac muscle will utilize free fatty acids to generate energy. Also, some of the free fatty acids may be found in the circulation, in the systemic circulation, and then albumin will bind to these free fatty acids and transport them to the liver, where it will be reused to synthesize triglycerides. Now, the glycerol that is also produced as a byproduct or as a product of the hydrolysis of triglycerides by lipoprotein lipase will not or cannot be utilized by these tissues, by these peripheral tissues. So they will flow in the circulation, they will go into the systemic circulation, and then they will be taken up by the liver, which can later be used to synthesize glucose through a process of gluconeogenesis. Gluconeogenesis. Now, this glycerol can also be used to synthesize triglycerides much later. After the hydrolysis of the mature chylomicrons, about 80 to 90% of the triglycerides will be hydrolyzed. So the major component that will be left in that chylomicron particle will be cholesterol esters, together with ApoE, ApoC2, and ApoB48. And that will result in the shrinking of the chylomicron. So once triglycerides is gone, remember in my last class, I said the size of the particle or the lipoprotein particle is dependent on the amount of triglycerides. So once most of these triglycerides have been hydrolyzed, it will result in the shrinking of the chylomicron particle. So you can see the size here, and then you can see the size, and the size is now smaller. This is now referred to as the chylomicron remnant. HDL will again associate with this chylomicron remnant, but in this case, the reason for its association is to receive the apoprotein which it donated initially in the cycle. From here, it donated ApoC2. So here it is back to take back the ApoC2, which will result in the formation of what? This. So ApoE and ApoB48 will now be left bound to the chylomicron remnant. The purpose of ApoE is for recognition. It is recognized by the specific receptor on the liver cells. So this receptor here is specific to ApoE. So what this chylomicron remnant does is it moves towards the liver, and on getting to the liver, this ApoE binds to its specific receptor. Once the ApoE is bound, the chylomicron remnant will now go into the liver through a process of endocytosis. And on entry into the liver, it will be hydrolyzed, and the contained cholesterol esters will be liberated into the liver. So basically, this entails the metabolism of chylomicrons.